Hi everyone, welcome to week 6 of your journey into the world of the unseen. Kind of on the home stretch now. The big takeaway from this week is that bacteria are not the only bad players in this game called life. There are a host, lots of eukaryotic pathogens as well, and we examine those in our study of microbiology. And many of those, while many of those are microscopic, of course, many of those are large enough to be seen with the naked eye, like tapeworms and pinworms and roundworms. We study those in microbiology as well. Sometime in the past, those pathogens got looped into study microbiology. So that's what we do. So you recall from your general biology studies, as well as readings that you've done for this class and for your lecture class, that prokaryotes, like bacteria, are organisms that do not have a nucleus. Remember that karyo pertains to the nucleus. Pro, the prefix pro, means before. So these are organisms that evolved before the nucleus evolved. While eukaryotes, remember, EU means true, karyo pertains to the nucleus, so these are organisms with true nuclei. So that includes things like amoeba, paramecia that you're familiar with probably from biology, way back in high school, um, and which are unicellular, but again, we also look at eukaryotic multicellular pathogens as well. So this week we bridge the gap between the prokaryotes and the eukaryotes by doing comparative surveys of a number of species of both. Now I suggest that as you progress through doing your surveys that you look for similarities between the pathogens, signs and symptoms of infection, and also treatment protocols. There are a lot of similarities between some diseases caused by bacteria and some diseases caused by eukaryotic, for instance, some protists. And some of those treatment protocols as well are similar. Some antibiotics work just as well against bacteria or just as well against some protists as they do against some bacteria. So look for those similarities. I provided links to 2 videos to help you understand the concepts here in topic 6. The first describes the basic procedure and purpose of ELISA. That's an enzyme that acronym that stands for enzyme-linked immunosorbent assay. You'll learn about that in your immunology lab. And the second is a brief introduction to eukaryotic microorganisms. My note here says that would be good to view before attempting exercise 13. Housekeeping for the week, working on topic 6, microorganisms and humans. Make your initial discussion post and substantive peer responses no later than Friday. That's this Friday, July 25, 11:59 p.m. GCU time. And remember, in order to earn full participation credit points, which is up to 20, you have to respond to 2 peers on each of 3 different days during the week. So the earlier you get started, the more you are able, the better you are able to participate in discussion. If you wait until Saturday or Sunday to do your responses to peers, that doesn't give them enough time to really interact with your response, and so that's not really the way discussion is supposed to work. So start early, work on it throughout the week, and you'll learn a thing or two. So you want to complete your pre-lab questions for labs. This is 8, 12, and 13. These are due no later than Sunday. That's July 27, 11:59. Now complete the labs. There are 3 labs this week, and they're all in Halo, okay, not in Connect. And there are some sheets, some documents that you have to download and fill in, and then that is what you upload back to me so that I can grade them. These are all three are due no later than Sunday, July 27. There's the immunology lab. That's the ELISA lab that I referenced a little bit earlier. Um, Now there is an image in Connect of the results of an ELISA. It's like 9 little tubes. Some are blue and some are clear. Use that. So because in your submission it says sketch the results of the ELISA. You don't have to sketch that. Sketches come later, but you don't have to sketch that. Just copy that image from Connect and paste it into your document, and then you answer the follow-up questions based on that image. Okay, you don't have to sketch that one out, right? But anyway, that's the ELISA, and then from exercise 12, that's the infectious diseases lab. So the instructions say describe the morphology via a sketch and identify the disease and describe the treatment for each organism on that worksheet. Now, those have to be sketches. You have to do those by hand. Some of you are able to, you have the little stylus and you're able to draw on your tablet or whatever and capture that image. Others of you may have to sketch the organism on a piece of paper, photograph that, paste it into your submission document. However, but it needs to be hand sketched. You cannot go to Google Images and find a quote unquote sketch of E. coli and then grab it and paste it in there, okay? You will lose points if you do not actually sketch these out yourself. So however you want to work that, if you need to contact me for clarification on that, please do not hesitate, okay? But you need to actually sketch those out by hand. And then exercise 13 is eukaryotic pathogens. And again, you need to sketch those out, okay? And then there are some follow-up questions on that, and then you submit those 3 worksheets, and one at a time. There's places for each one. And that's how you earn your grade for those assignments. As a reminder, now remember, if you have not already turned in I haven't checked yet, some of you may have, but if you have not turned in your benchmark assessment, remember that's your lab report that you started 2 weeks ago, it is due Sunday, July 27, 11:59 p.m. No extensions because you've had 2 weeks to work on that. So make sure you get that turned in if you haven't turned it in already. And here's a heads up, your final exam next week is the last week. Your final exam is next week. It's comprehensive, starting from topic 1 all the way down. I posted, remember, a review of the first 3 weeks in video form. I suggest you go back and watch that to remind yourself. Those topics will also be on this exam, and I'll post a review of topics 4, 5, and 6 for you this week as well. Pay particular attention to due dates on all of your assignments. My note says I have added an optional DQ. I haven't added it yet. I'll do that as soon as I get through here for earning participation points. Remember, on my prompts, your post must be substantive, but you do not have to reply to any colleagues unless you want to, and that post is due no later than Sunday as well. Also, I want to give you a heads up. Next week, you will be doing a presentation, not completely different from the one that you're doing for lecture, but you will be doing a slide presentation, so you might want to look ahead and see what's going on about that. More about that later. And I have a link to a video about the gram stain. No, wrong class. But anyway, so if you have any questions or comments or problems, contact me as soon as possible. Message 804-229-7993. Use your Halo inbox or message box. Certainly, you can use campus email. Two quickest ways to get a hold of me are message and Halo. All right, that's all I have for now. Let me know what's going on. Have a great rest of the weekend. I'll be back with you more this week as well.